I'm Erica Pimenta. I'm a physician scientist that's focused on sarcoma biology. And one of the first studies that we've accomplished and finished is trying to understand what is the molecular basis for the two phenotypes we see in liposarcoma, well-differentiated and dedifferentiated liposarcoma. And this is an important question because it's a fundamental question across all cancers, like what causes dedifferentiation. So we were excited to tackle this question in the liposarcoma space where new treatments and new understanding is really desperately needed. So we designed a cohort of patient samples since liposarcoma doesn't have a really valid in vivo model system that recapitulates human disease. And we employed multi-omic single nucleus sequencing, and that lets us look at both the epigenome and the transcriptome at the same time from every nucleus within a patient sample. And in doing so, we got a global epigenomic and transcriptomic view of both types of liposarcoma, and we were able to pinpoint really loss of this pro-adipogenic signaling pathway in dedifferentiated liposarcoma. And then the really fun part of this project was we could move into in vitro models to perturb that pathway more carefully. And we actually found that PPAR gamma isoform 2 is the molecular switch between the liposarcoma phenotypes. And so I can now say that our conclusion is that PPAR gamma isoform 2 is missing in dedifferentiated liposarcoma. And when you add it back, you get this well-differentiated phenotype in vitro. We then became really interested in how that is regulated, like what shuts down PPAR gamma isoform 2. And for that, we could go back into our ATAC sequencing or epigenetic human data. And what we found was that epigenetic silencing right at the promoter of PPAR gamma 2 is probably responsible for this shutdown. And that opened a whole new avenue of research in liposarcoma because the epigenome became really important, became this huge focus. And so looking globally at the epigenome in well-differentiated versus dedifferentiated liposarcoma, we actually found that dedifferentiated cells are poised for GLP-1 signaling, which really piqued our interest. And so we are now undertaking studies to try to understand what GLP-1 agonism can do in dedifferentiated liposarcoma, as well as in normal preadipocytes, which is an area of research that is understudied but in great need given the rise of GLP-1 in the population.